we're going to start. Hi, everyone. I'm really pleased to welcome you and welcome Dr. Carika Weldon. I hope everybody in the Zoom can hear us clearly. I will make a short presentation of Dr. Weldon, and then she will tell us about nanopore sequencing and its possibilities. So Dr. Weldon is a Bermudan biochemist and founder and CEO of Carika Genetics, the world's first Caribbean-based genomics company. Prior to this, she was a lecturer at the Montfort University and founded the Bermuda Principles Foundation. She worked as the Oxford Nanopore project manager at the Oxford Genomics Center at the University of Oxford in the United Kingdom. And in April 2020, she was called by the Bermuda government. She was appointed as the science advisor to the premier and laboratory director of the government COVID-19 lab, which she created. Dr. Weldon was granted a Queen's Certificate and Badge of Honor for this work. So please welcome Dr. Carika Weldon. You can now start your presentation, which will follow a brief session of questions, which you will allow it. I will put on your slides, and you can start whenever you're ready.
Perfect. Thank you, everyone, for coming. I'm so sorry I don't speak French. All I have is bonjour. That's it. I'm really excited to be here to talk about nanopore sequencing, which is a technology that we use at Carika Genetics that we are actually going to be bringing more to Guadeloupe, very excitingly, and I want to share the possibilities that it has. Okay, there we go. So just a bit of history about nanopore sequencing. So it was actually first thought of as an idea in 1989, which was the year before I was born, actually. So my whole life has been about nanopore sequencing. And the idea was that a protein, so a transmembrane protein, can actually fit a DNA strand through it, right? So that was the first idea. In 1991, the professor discussed it with another professor at Harvard, which led to them wanting to move it forward. And they had initial experiments in 1993, with Professor George Church, who is one of the leaders in genetics. And he joined the team, and they published it in 1996. 12 years later, in 2001, they then decided to actually bring it to life as a company. And so in 2005, they actually created Oxford Nanopore Technologies. And the first sequencing device was actually presented in 2012, so not long ago, at a conference. And that's when they introduced the MinION, which you have at the Institute Pasteur. And it was launched for people to start using it 10 years ago. So relatively new technology. Since 2014 and onwards, it's now in many countries around the world. They have more machines I'm going to talk about, and there's lots of papers. So it's becoming more and more popular as a technology. So how does it work? Just to clarify. So all of the Oxford Nanopore sequencing, I want to read it. Yeah. All the Oxford Nanopore sequencing devices use flow cells that contain these tiny holes or nanopores, and they're embedded in an electro-resistant membrane. So it's an artificial membrane with an artificial nanopore. And you can see here, that is representation of the pore, and you can see it's just big enough for one strand of DNA to go through it, right? Each nanopore corresponds to its own electrode, and it's connected to a channel and a sensor chip, which measures the electric current that's going through that pore. That's the full basis of what's going on. So I like to give the analogy, it's like water through a pipe, right? So water is flowing through that pipe, and each time a different base. So you know with genetics, you have 4 bases, A, C, G, T. Each time a different base goes through the pore, it perturbs the electric current differently. So if you have water flowing, you'd have the A that would make the water look different to a G, to a C, to an A. And so that is how it reads the sequence. It gives what we call the squiggle, which I'm going to show you. So it's all the electric current, and it uses that to convert it into the bases. Okay, so again, so you have the pore, and you can see on this side here that you have the electric current, and at an A, it's here. A T, it's in a different place. C is different to a G, okay? So it's converting the electric current into this, and then it's converting these electric readings into actual bases. Now, the other interesting thing is an A by itself is not the same. It depends on what came before it. So if it was a C before an A, it looks different if it was an A before an A. There's a lot of combinations, as you would understand in this department, lots of mathematical permutations. And so there had to be a way to like figure out all those combinations. And so they use machine learning and AI. So it's using what it's learned to convert the squiggles into the bases in real time. Okay, so this is the full process that I do in the lab. So we get a sample, it could be a swab. That's a COVID swab that we all didn't like getting. Or you can get a blood sample, or you can take a cheek swab, or you can take mosquitoes, or you can take bacteria, you can sequence anything. You're gonna extract the DNA from it, and then you have to prep the DNA to be to go to the pore. So there's some chemical reactions you have to do. And then you can see here, the sequencing, and it's giving you the squiggle, and then it's converting that into the bases here. But then you have to assemble that back, right, to make sense of it, because it's going to give you pieces. And so that's where you have to have a reference genome, or if you're making a reference genome, you make a assembly. Okay. So the beautiful thing about nanopore sequencing, as you know, you have A, C, G, T, or U if it's RNA. Usually when you do sequencing, has anyone in here done sequencing before? Yes, okay, good, yeah, one person. So usually when you do sequencing with other technologies, you have to do PCR. You know what PCR is, right? You have to amplify using PCR. But what that means is that you're losing some of the information that's on the DNA, the methylation or whatever other information is there. So the good thing about nanopore sequencing is you don't have to do that. You can do without doing PCR, and you can sequence the DNA natively. So that's what I have here. So you can do native DNA or RNA sequencing. So you don't have to lose any information. All right, so have you heard of Illumina before? Yes, you have. Anyone else?
No.
Yes? No?
No.
Okay, so basically the first way to do sequencing, the main company that sells it is called Illumina. It's short read. You have to break it up into pieces of 150 base pairs. You do PCR. And it's using a different chemistry. So that's the main technology that's used. But if you look at my puzzle here, it's like breaking it up into all these really small puzzles, right? Who wants to do that? Anyone like puzzles? I don't like puzzles. So the issue is, because you're looking at 150 base pairs at a time, it gets really difficult to look at big deletions. If a deletion that's 4,000 bases is missing, you can't see that. You don't know where it starts and where it ends. If you have an area in the genome that repeats, so it could be like 100 A's or 300 A's, then you get lost. You can't see how big it is. So this is where now we have, that was short read sequencing, this one here, don't like it. Then we have long read sequencing where we can sequence bigger pieces. And the good thing about Nanopore is that it will sequence whatever size you give it. You don't have to break it up into those small pieces. There are some people who sequence super long pieces of DNA, talking about mega bases long of DNA. So they're like very special group of scientists. But on average, when we're doing human genetics, we tend to do around 15 to 20 kilobases. So 15 to 20,000 bases at a time. And then we can see big deletions, and we can see if it's inverted, and we can see those repeats and see them very clearly. So it's like having a puzzle, but with bigger pieces. And it's now easier to put that back together as well, right? We were just in the cluster room. So you're gonna need less compute power to put this puzzle together versus this one, right? Make sense? Okay. So one of the beauties as well is that there's flexibility with Nanopore. You don't just have to have a super big machine. You don't have to just have a small machine. They have a range of products. I know that from speaking to David at the Institute Pasteur, they have this one, the MinION, which is the smallest one. And then it goes all the way up to the PromethION, which is the biggest one, okay? I'll start going through some of these so you can see. And I'm happy to provide the slides as well if you want.
Okay.
Yeah.
Nice.
So you don't take pictures if you don't want. So MinION is the smallest one. I actually bought one of these. I have my own personal one in my room in Bermuda, my own personal sequencer. It fits in the palm of your hand, very tiny. And it costs about $2,000 US. So that's why I bought my own one. And you literally can, like, it will fit in here. You can literally travel with it. They had this one in the middle, this one that had the computer with it, but it's now no longer around. And this is the newest version, the MK1D. Okay, so you connect it to a laptop and you run it on the laptop. Your laptop has to have enough power, has to have the right specs. You guys know about all of that. But you can do sequencing anywhere as long as you can take your laptop. The laptop actually powers the MinION, so you don't need to plug it into a wall. So you can actually do sequencing in the field, wherever you are, as long as your laptop has charge. Yeah. Okay, this is the next one. It's called a gridiron. You can probably see there are basically 5 of these. It's almost like 5 MinIONs in one. Okay, so it uses the same flow cell as a MinION. Most times I've heard people having this are in clinics, or if instead of buying 5 little MinIONs, you have, this gridiron. It's quite popular and is actually getting approved for clinical use in hospitals now as well. I don't remember the exact price. Don't quote me on this, but I think it's around 30,000 US, around about there. Then you have the biggest ones. So these are the PromethION. So you actually have a range of these as well. The difference is that the flow cell where you load the DNA is actually bigger, and I'll talk about that in a minute. So you can get a lot more DNA data at once. This one is the absolute biggest machine. So it's called a PromethION 48. It has 48 positions to run the PromethION flow cells. You can do 48 human genomes at once in about 3 days. It takes about 3 days per human genome. In Bermuda, where I live and work, we have this one, the P24. And they've come up with these smaller versions so you can have the same power to generate the data, but it doesn't have to be as big. So you have P2s, but these are the newest ones. This one doesn't have the computer, the P2 solo, and this one does. So you can have it with all the compute power as well. I want to say this one is about 60,000.
60?
Yeah, 60. And I can't remember the price of this. I think it's over 100, but it comes with the computer, so that's why. Okay, and then we're gonna go back. So even smaller than the MinION, there's something called a flongle. So normally all of these flow cells you have to send back to Nanopore because it's like it's got all the electric stuff on the flow cell. You have to send it back because of that reason. These ones are disposable. It is about 1/10 of the size of a MinION flow cell, and you can just try out to see if your data or your sample looks like it's gonna work before you run it on a big flow cell. I think these are really cute. I have a few of them. And you can see the typical data output is like 1 to 2 gigabases. So if you're talking about viruses or bacteria, this works pretty well. You're not going to do a human genome with these. That would not work. Okay, so more about the flow cells. So I'm going to focus on the largest one. So this is the flow cell for the promethine. It looks like a square, exactly like that. So it contains 2,675 channels. And each channel has 4 nanopores, right? So in total, you have 10,700 nanopores in that one flow cell. The difference between this and the MinION flow cell is instead of having 2,675, it's 512. So it's about 5 times smaller. Yeah. When I see something in the chat, are you monitoring that? Okay.
Yes.
All right, this is where it gets a little technical for me, but I figured that you guys would like it being in this department. So the wells are inserted into an electrically resistant polymer, as I mentioned, by an array of microscaffolds connected to the sensor chip. Each channel is associated with a separate electrode and is controlled and measured individually by the application-specific integration circuit. I feel like you guys would love that. ASIC, that makes a lot of sense to you, I'm sure. Yeah. This went above my head. I'm not technical like that. But the beautiful thing for me is that I can see what's happening in real time in each pore and know what's happening. Okay, so one of the things that I really am super duper excited about is something called adaptive sampling. So as I mentioned, the pore is big enough for DNA to go through it. What I didn't mention before is that inside the pore, there is a probe that is where it's reading the electric current. Now, what you can do is you can actually give the machine a bed file, which is the file that it reads which region of the genome to look at. And normally what happens is it would just sequence anything. But if you give it some instructions, it will, up the top, you can see, it will start sequencing. And if it matches what you told it to read, it will keep sequencing it. But if it starts sequencing and it's not matching the region that you want, it spits it out. Pretty cool. So now you can get enriched sequencing all on the machine. So from my side in the labs, I'm a wet lab scientist. Usually, when you want to do targeted sequencing, you have to do a chemical pull down, you have to have probes, you have to order it from the company, and that takes time. It takes 2 days to get DNA and then pull out the genes you want and then sequence it. With this, it literally takes 5 minutes to tell you what genes you want, and it does it for you. So some of the applications of this particularly is the scientist that actually invented this. He's from the University of Nottingham. His name is Matt Luce. I was in a meeting with him, right? He's on our sequencing service committee in Bermuda. And he's on the meeting on Zoom. He's like, I'm looking at the screen. We're looking at someone who's in brain surgery, who had the biopsy taken. We've sequenced it. We know what mutation is driving their brain tumor rather, and they're still in brain surgery. Like they figured it out whilst they're still in surgery because it was that quick. It only takes like 4 to 6 hours. The brain surgery takes a lot longer, sometimes 20 hours. So we can be really quick with how we can tailor what we do with genetics now when it comes to clinical, at least. Right now, we use this in our lab to do cancer panels. So if someone says, they want a cancer panel, we can tailor what genes. One of the things that I'm really excited about, we are going to be doing a prostate cancer study. We're doing it across multiple islands in the Caribbean, but we're doing it in Guadeloupe and French Caribbean as well. We can actually add genes that are relevant to the local population into what we're testing in 2 seconds, rather than having to send it to a company to get primers and take very long. We can also do non-cancer. So we have a clinical lab, a genetics lab in Bermuda. We have to do sickle cell testing. I will do that during adaptive sampling. We can literally tailor it for whatever gene we're interested in. Okay, so data analysis for this. So again, I think that's a repeat of the slide. That's what I want. So applications, there's a lot of things that we can do with this technology. I'm not going to go through every one of them, but I'm going to try to touch on as much as I can. They are broken up into 3 different categories. So you have basic research, which is all of this. You have clinical use, which are all of these. And then you have on-site applications, I see you're squinting, which is these. And there are 3 main kind of features of nanopore sequencing that make it very unique. One is that you can read those long pieces of DNA. You can read DNA without having to do PCR, right? Remember that? And then it's portable, right? So you see some of these are basically showing how it's basic research and how it's useful to have those different features. I'm going to go through this bit by bit. Okay, so the first one is closing the gap in reference genomes. We know, I want to say in 2003, when the first human reference genome came out, it was all done by Illumina sequencing, so short read. And this is actually one of the main uses of nanopore sequencing is, that thing's in the way, but yeah, 30% of published papers of nanopore is doing this. And we know for the human reference, for example, it was about like 90% complete, but there was the telomeres that we couldn't read through. And so by using the long read sequencing, we were able to finish it. And I think that came out in 2022. Now we have telomere to telomere sequence of the human genome. All right, we can build new reference genomes as well. So new genomes that are required. I use this one because you all know about it. So the first genome of COVID-19 was done using nanopore sequencing. And because we now had the sequence, we can obviously make the test and everything. And we can actually do genomes that are quite big. So you can see some of the bigger genomes that have been done are 34 gigabases. That's 10 times the size of the human genome, right? So you can do quite big. I know for us in Bermuda, we're actually going to be doing our national tree. So it's a cedar tree, and it's expected to be around 12 gigabases. That'll be one of the biggest genomes that's done with just nanopore. Yeah. Obviously, from a compute perspective or informatics perspective, you need a lot of data to make a new genome. We tend to want to have 60x coverage. So if a genome, we want to do mosquito, right? We want to do mosquito. So if we know the mosquito genome is 1 gigabase, then we need 60 gigabases of data to then put it all together. It's a lot more challenging to do this because you're piecing it together for the first time. But this is your specialty, right? This is bioinformatics. So you guys love it. And just to give some scope, so only 3 PromethION flow cells were required to sequence a de novo human genome, and it took about 6 hours of computational assembly. Again, because you have big pieces, so you can put it back together quite quickly.
So entirely de novo, no reference. Yeah,
so this was actually done. The reason you do a de novo human genome, and we're going to be doing that too, actually, with you guys, is the reference, as you probably know, is biased. The reference genome that we're using for any type of variant calling is not representative of the diversity of the world. So we're actually going to make a Caribbean reference genome that we can then use, and we're actually looking to make a Guadeloupe one. You can actually go even deeper. So for this one, I think it was a Pan-African genome that was done. They took about 1,000 people of African descent and then made a new genome from scratch. And they actually found 10% more genetic material than the reference. So you've got to figure out what that all means, but that's useful. But yeah, so de novo. Then you have novel variant identification. So because you have the long reads, you can actually look for different variants that you haven't seen before. We actually experienced this. We did a breast cancer study in Bermuda. We did everyone with long read sequencing. And because we can see, big chunks, in a cancer gene, there was like a SNP, so a single nucleotide variation, and a structure variation kind of like compounded on each other. It was very difficult for the short read sequencing to see it, but we were able to see it very clearly. Initially, they told us that it was fake. They said, "Oh, your sequencing is making up stuff." I said, no, it has to be real. And we sent it to another lab, and they saw it as well. So we can actually find these new variants really nicely. I am an RNA scientist, so I do genetics, but I am a transcriptomics person. So really excited about this one. So you can actually use it to look at the full transcripts. Because again, usually you have to break it down into 150 pieces. But now you can look at the whole transcript. Transcripts are not usually that big, but some of them can be quite long. And then you can look at the alternative splicing as well to see the different variations. All right, this one is a big one here in Guadeloupe with the research you guys do, so infectious disease. So you can do stuff in real time, which is very, very important. And again, being portable, you can take this in the field or wherever is very important. So it's been used for rapid pathogen detection, including diagnosis of meningitis, bacterial lower respiratory tract infections, infective endocarditis, pneumonia, and infection of joints, right? So obviously those things are, time is of the essence in those diagnoses. So this is an example where 16S amplicon sequencing took only 10 minutes with the MinION. And the cool thing as well is that you can stop it when you have enough data. You don't have to just keep running it just because you started. Once you have enough, you can stop it and do your analysis. And yeah, another example, MinION was used to identify 51 acquired resistance genes directly from clinical urine samples that usually you have to do cultures of. That takes obviously more time, right? So you can get to your answer a lot quicker. And again, it's mobile, so you can take this into the field, which is very useful. All right, structural variations, like we talked about earlier. So because you have these big windows that you can see at once, you can start to see deletions that you previously didn't know were there. We have picked up a few of them as well in Bermuda. We're looking to do that more in our Caribbean genome program. So I haven't mentioned, but we are looking at perigenetics to do 100,000 Caribbean genomes by the end of 2027. So that's a lot of analysis. So we'd love to have you on board if you would like. So speak to David if you want to join us on that, but we will be doing all of the French Caribbean genomes here in Guadeloupe at the biobank. So we'll do sequencing here. But yeah, structural variations is becoming more, well, actually the first cancer genetics was shown to be a structural variation, but it's becoming more clear that we need to stop just looking at SNPs for genetic, cause of disease. And because if you delete a whole section, then the whole thing's gone. That's also going to cause disease. So you can see that very clearly. Okay, when you're talking about more rare genetic diseases, that's also where this comes in. So an example is our nanopore sequencing of human genomes revealed an expansion of those tandem repeats that are linked to Alzheimer's. So again, remember I said, when you use short read, you can't see how many times the repeat is happening because it all looks the same. You can't, like, is it one or is it two? You can't tell. But if you can see the whole thing at once, then you can actually say how big it is. And so that was something that was found in this ABCA7 gene that now is associated with Alzheimer's disease. The other cool thing that we're going to be doing as well is using nanopore sequencing for prenatal testing. So you can check that the baby is healthy and has all the right chromosome numbers, doesn't have any aneuploidy, and you can do that in 4 hours versus 1 to 3 weeks. So again, speed is important and getting an answer quickly. All right, this is where our beloved COVID comes in. So outbreak surveillance, you can use the MinION, and it was used quite a lot for monitoring COVID very quickly. In Bermuda, we didn't have any sequencing capacity when that came, but we had a MinION, so we were able to start doing it. You can see here that in April 2015, the MinION devices were shipped to Guinea for real-time surveillance of Ebola outbreak. You only needed 15 to 60 minutes per sample. So again, time is of the essence. And again, you can see that used for surveillance of Zika virus, yellow fever virus, and dengue virus outbreaks in Brazil, which I know are things that are studied here in Guadeloupe as well. So again, it's about having an answer quickly and doing it in a more efficient way. Metagenomics is also very, very useful. So this is identifying different microorganisms. And this doesn't just have to be clinical samples, it could be biodiversity samples. I know we had a chat about that a few days ago. We actually have a project in Bermuda where we're going to take ocean samples, so ocean floor, like soil samples, and sequence it and see what bacteria and microorganisms live in there. Because they secrete chemicals that might be potential therapeutics. So we can do that sequencing on Nanopore very, very easily and see what's there. Again, difficult regions of the genome, because we can see it in one go, we can actually figure out what's going on as opposed to having a very limited view. We talked about this a bit, but those repetitive regions become a lot clearer, and hence why we can complete those genomes. And my favorite, epigenetics. So, again, you have genetics, but there's also that chemical layer on top of that, which is there's modifications on both DNA and RNA. And every other technology, because you use PCR, even when you're doing RNA sequencing, you usually have to convert that to cDNA, which is the DNA version of the RNA. So you lose all that information. But this is actually how I got into Nanopore, because I was actually using a modified guanine in my PhD. I wanted to see if I can see it. You can actually see this information. You don't lose it, you have it. So actually, our breast cancer study, we did 102 women in Bermuda. We have their whole genome and their whole epigenome. That's the first data set ever to have epigenetic data on Caribbean people. And as we do our prostate cancer study, which would be 1,000 men across the multiple islands, we're going to have epigenetics as well. Everything we do at Carigenetics will have both, so that we can understand. And why is this important? Because the epigenetics is interacting with the environment, right? You can have someone that lives here in Guadeloupe or someone that grew up in France, different environments, and you should be able to see that in the epigenetics. We can start to look at that, right? And the other thing is that epigenetics can be reversible. So if you find something, you can try to fix it. You can turn it on or turn it off. All right, and we said this, again, you can sequence in the field, you can take it, literally, I have sequenced myself in a hotel room. I did. I confirmed I was human. I had the right amount of pairs of chromosomes, and I confirmed I was a female, which was actually very daunting, to be honest, because I was like, oh, I have a Y chromosome to pop up. But yeah, I literally sequence anywhere, which is very, very useful, especially during biodiversity work. We want to go to the site or surveillance. Okay, almost done. 2 more slides. So some advantages of doing nanopore sequencing is that, again, it's in real time and you can scale it as you go. Actually doing the library prep or the preparation of the sample for the midline, exactly the same for the PromethION. So when I went to Oxford, I had only run a MinION, and then I became the project manager of all the big machines. I was like, oh, but it was the same, so that wasn't too bad. So it's really good. You can start on the small machine and get competent and then go to the bigger machine. It's very compact and portable when you have the smaller one. Again, you can go outside traditional lab environments. It also contains devices for rapid laboratory preparation. So they actually have a machine that will prep the sample for you as well, if you want. I'm old school, I wouldn't prep it myself, but you can buy that as well. There are different parameters in Oxford Nanopore sequencing, like experiment duration, device use, that can all be adjusted. Like you don't, again, you're not fixed into, well, I press start. It's like when you do your laundry, right? If you start the laundry, you gotta wait till it finishes. You can't like take it out early, it's a bit annoying, but you have to wait. But this, again, you can track how much data you're getting and then stop it when you have enough. And there's flow cells I mentioned, you can wash them and reuse them, which no other technology can use. We are very good at that in Bermuda. We wash everything and we rinse it till it's done, because you get your money's worth. Let me tell you. Okay, and the Oxford Nanopore sequencing devices can sequence long fragments, so you can see what's going on. It makes simplifying genome assembly, and you can sequence directly the DNA without PCR amplification. The other thing you have is bias for PCR. It prefers certain bases, so you can get rid of all of that. Now, that could be measured. There are some limitations as well, so we need to know those. So there is a need for large amounts of input material. So if you're doing no amplification, that means you need more material to start with. So for our human genome work, we usually need a microgram of DNA. Yeah.
What does it mean in terms of blood sample?
So what we do, so that's the question. We tend to take 4 mils of blood. We only need half of a mil to extract. And we tend to get, my team are doing pretty good. We tend to get about 20 micrograms from that. So we don't need more blood. It's just we need, human is all right. It's more so the RNA input, which is a bit difficult. You need, again, like a lot. And if you're looking at like FFPE samples, very difficult to get DNA out of tumor tissue that's fixed. So it's difficult to do the sequencing. Yeah.
The paper, the blood, store on paper, that's the paper count.
Oh my God. So someone I met, I can't remember who it was, in London 2 weeks ago, they actually can extract enough blood from a blood spot. I'm super excited about this. Because then you don't have to be getting phlebotomists to draw blood. You just get people to do your blood spot and then you extract it. Or you can get enough DNA apparently from that as well. It would be more on the limit. I like to have more than I need so that I can feel comfortable. So that's why we draw blood, but you should be able to get enough from that as well. All right. Nanopore sequencing can be complex to analyze, and you need people like you that know what you're doing and bioinformaticians to use it. Mind you, they do have a software called Epitome that you can install on the machine. So like right now, I'm not a bioinformatician, but I do all our variant calling for all our samples because the software does it. So they are getting better. It has a lower throughput, so you can do 48 samples at a time, whole genome, but that's your limit right now. You can't, unless you buy like 10 machines, and then you can do 500 at once. That's gonna cost a lot of money. I mean, the difference as well is that you have, because it's longer pieces, it generates a lot more data, and you need a lot more storage. So with our breast cancer study, we did all the sequencing in 2 months, and we have a data center in Bermuda, and we maxed them out at 100 terabytes in like a month. So we're working on that. We're talking about having like storage here for when we do all the genomes. But you can't be doing like 1,000 genomes at once unless you put a lot of money into it. That's a bit of a downside. But again, it's because it's longer. And it can be very sensitive to sample preparation and contamination. So it is a high skilled job to handle these samples. I mean, sciences in general, but it's gonna pick it up. Like even for us, if you don't shear the DNA, so you have to kind of break the DNA up when you after you extract it, so it's a nice size. If you don't do that correctly, it messes up the pore because the pore gets clogged and it's not as efficient. So you have to be train people very well, and they have to be on point when they're doing it. All right, that's it. We made it.
Thank you so much.
Any questions?
So far, there are no questions in the chat, but maybe in the room there are some questions.
Yes. So just a question because I'm not sure I understood.
Yeah.
Okay. How do you measure the mistakes if there are any mistakes?
Good question. The error rate. So I didn't touch on that because it's a big controversial. So when Nanopore first came out, it had a really bad error rate. But they've gotten better. So it's now at a good place where it's pretty accurate. What we do in our clinical testing, because if you have a variant, you need to know if it's a real variant versus the machine making up something. I actually look in IGV, like I actually look at it in the genome viewer to see like what's happening. So we're actually going through a validation right now. We have 48 samples that we got from another lab that uses nanopore sequencing, and they do clinical testing for cancer. And they already know what the result is, and we're now testing them. So I've been doing that a lot because we have to like get the right answer. But for some of them, it's really clear that, yeah, this probably was a mistake versus it actually being a mutation. So you have to look, I mean, there are on the informatics side, there are like some softwares that look at the error rates. I have no idea about how this work because I'm not that side of it, but there are some programs to run. But it is something to be mindful of when you do sequencing, especially when you're diagnosing something. You don't need to tell, oh, you have a mutation. It was just the machine doing its own thing. Yeah. So we tend to have like a cutoff. I wonder if I can show you. Want to see the machine? I can log in remotely from the machine as well. Very exciting. I know it's a thing. Give me 2 seconds. Let's see if I can bring it up. I'll keep talking as I go. But yeah, we have a cutoff. I need to see, well, first of all, we have to have enough coverage. That's the first thing.
What's the limit in general?
So the clinical number is 30x. So you have to have passed that location 30 times and feel confident. What we're actually finding is we can go as low as 15x.
So 13 for clinical.
The clinical is definitely is 30.
For research, 15.
Depends.
Well,
So like we did genomes of our national bird. We did a reference genome already, and now we're doing it of I think we did like 42 of them now. And because you have 42 of the same species, you can have like a pan genome. So we don't need 30x of each one. It all kind of compounds. So yeah, it depends on what you're doing. But 30x is like the gold standard for clinical. I'm just logging in. Exciting. Okay. I think I have an IGD. You know what IGD is? Integrated Genome Viewer. You know what it is? Okay. Log in. Any other questions while I get this going?
so far in the chat, there are no questions.
Okay.
Just while you're connecting, I'd like to be sure to understand what do you put exactly in the device? Is it blood or is it something else?
Oh no, no blood. No. So we put in pure DNA that has been chemically prepped to go into the flow cell. It has to have sequencing adapters that like kind of draw it to the pore. So you have to prep it. You can't just, definitely no blood, definitely no, there. Anyone in my lab did that, I would be like, oh. No blood. So you need purified DNA that has been what we call library prepped. So it's a chemical reaction, takes about 2 hours. And once you have purified DNA, it takes about 2 hours to prep, and then that's what goes in the machine.
Okay.
Yeah.
And in Bermuda, what kind of human diseases did you start to look at already?
Breast cancer,
Uh-huh.
prostate cancer.
Prostate cancer.
Yeah, so we're actually recruiting now. We have 47 men that we've already got their blood. And we've done a PSA test as well. Okay, I think it's up now.
So you are looking at somatic gen mutations.
Just germline for everyone, and then if they have cancer, if we can get the biopsy, we'll look at somatic as well. And then we're also doing some sub-studies, which are RNA, so transcriptome, and then ctDNA, so that's the cell-free DNA. So the tumor itself secretes its own DNA, we can pick that up as well. Yeah. This is our machine, honey, though. All right, so you see we have 24 positions. And I operate this, I literally do this from home like all the time. All right, so this is IGB. I'm gonna load a file. Let's see if I remember which one. I want to get a good one. Do you want to see a deletion or a SNP? I got options. A deletion? Ah, okay. Deletion coming right up. All right, 1 second. Okay, this was a nice one. This is the first one we did actually. So, number 1. And let me just show you. So, this is epitome. All right, so this is where the bioinformatics tool for Nanopore is on our machine, does all the analysis on the machine. And we have different workflows. So this is the one we use, human variation. So it looks at SNV calling, structural variation, modified base, CNV and STR, but there's other ones you can do as well. If I go here, this is the, sorry, the internet access is not as great. Okay, so just to show you, this is the alignment report. So it will tell me what the coverage was for this sample. So you can see it was 12.194x. So it wasn't 30. Didn't have enough data for 30. But I wanted to see if I could see the SNP properly. So in the SNP report, this is from Epidemi again, and I did all this myself. So no bioinformatician. The machine did it, but yeah. So in the SNP report, you can see it's telling me there's, too far. Yeah, you can see it's telling me there's a pathogenic deletion in BRCA1. It's telling me what position it is. It tells me the consequence, and then it's giving me, telling me what's happening in which transcript it's in, and then how it affects the protein. Right, so that's all output that I just did myself without, just pressing buttons. Then for me, I want to look at it because it's 12x, it's not 30x. I'm like, let's see if it's real. So, wait, load this up. Just gonna load the BAM, what BAM files? I'm familiar with that? BAM files. So BAM files are the file that has the information of the reads and has it mapped to the reference, so you can see it. All right, so this should have the BAM file loaded now. Yep. So this is so these lines here, these gray lines, those are the actual sequencing reads. If I click one, it should actually, it's telling me which read name it is and how long it is and all that type of stuff. Okay, so let me find BRCA1 first. So I can just type the gene name in there, it'll take me right to where the gene is. If I need to get the exact correct location, we get that. 43094. All right. 094 16. See a deletion? You see? And let me just add this on top. This is what I do as well. I would add okay. So where that blue is, that's the SNP it told me about. And I can clearly see the deletion. It's very clear. Number one, I can see this. This is telling me like the coverage. You can see this whole way through its half. I can click this and it will also tell me what the numbers are. So it's saying it's 5 that don't have the deletion and 8 that do. So it's clearly there. So even though it was only 12x, very confident that that's a deletion in this person, that they have this mutation, right? So to answer your question, like, that isn't the machine making it up, right? Like, for it to keep deleting it in that one place every time, like, that's clearly something. So that's what has to happen when we do sequencing. We have to look at it in context. Now, if this was a region that was repeat, so the sequence matters, if that was like all A's, then I was like, maybe we should get more coverage, but it's not a repeat region. So very confident that that's the pathogenic variant that was causing their cancer. And the reason why, do you know why it's causing cancer? So to convert your sequence into the protein, it reads it in chunks of 3, called a codon. So you can see at the bottom here, you see these letters? They're in chunks of 3. Three letters, and it's giving you a different letter. So three of these sequence letters, and it's giving you a blue letter. Those are the actual amino acids that make up the protein. Because they're in chunks of 3, if you ever delete or insert something that's not a factor of 3, that means you throw off the whole sequence after that. So that's what we call a frame shift. So instead of reading it in 3s, okay, fine. Because if you just take out one amino acid, or if this was 39 deletion, then you take out what, that 13 amino acids, that might still work. But because of the fact it's not a factor of 3, it's now ruined how you read those sets of 3. So the rest of the protein is now wrong. And now it's not gonna give you the right structure, it's not gonna function properly, hence. And the BRCA1 is a DNA repair gene. So it actually helps broken DNA get fixed. So if it's not fixing broken DNA, you have more DNA that's getting broken, and then you have cancer. Very long way to answer your question, but very good. Any other questions?
Not in the chat so far, maybe. Nothing, yes?
I won't have any plan, fortunately I have to leave, but I'm a clinician, so
Okay.
no idea of what it was before, so
Yeah.
it's very interesting.
Good.
Clearly, I believe that this will change a lot in our usual way to look. We have to keep in touch.
Yes.
I'm from Martinique, I'm not from Guadeloupe,
Perfect.
but
Okay.
we have same needs, either in clinical management of the patients, but in clinical research also. So we have to work with you.
Yes.
Both you all guys. So that's very interesting.
Yes. Actually, for the prostate cancer study, we're working with Dr. Laurent.
Yes, no, absolutely. I know the guy.
Perfect. So he's the main urologist.
All right.
And so we are actually including like we're including Martinique and all the other French Caribbean islands.
But Laurent, he's a prostate guy.
Yeah, yeah. So this for the prostate cancer study. But as we do more, so what type of clinician are you?
Cardiology.
Okay.
Yeah.
Okay. So we want to definitely keep in
And
touch.
we have a lot of questions from this point of view.
Yeah. Actually, we did a cardiomyopathy genetic test the other day in Bermuda.
Okay.
So
What is it?
it was basically someone that had a little cousin that was 18,
Okay.
died from, I can't remember the exact, it was a cardiomyopathy, I can't remember the exact name, but he had a mutation in a gene, MYC-PB3, I think. And they wanted to know if he had it as well, so we tested him for it, he didn't have it.
Okay, okay. It's interesting because we are right now doing a research in Martinique for something like 70 patients with cardiac congestive heart failure. What we found was that maybe 20 to 30% of these had a mutation, but with the usual way of doing things. So
right.
Maybe
there might be something else.
Exactly.
Yes,
This
yes.
is something that we do very easily.
Yes, 100%. Would you be willing, because we're going to set up a sequencing hub in the biobank here in Guadeloupe? Would you send samples if you're comfortable sending?
Definitely.
Any more question? Busy date. Probably yes, but not for human, but for things.
That's fine. We need biodiversity as well.
So is there a lot of capability to multiplex samples also if you don't want? So I will probably not be interested in having one equivalent of human genome per flow cell.
Yeah.
Because that's the thing. You load one sample or one flow cell.
Yeah.
Like you have the genome.
Yeah.
But now for animals generally or different it's like 1/10 of the human genome or something like that.
Yeah.
So I thought that what's the idea is you can put some barcodes, so we got typers
Yeah.
before, so you extract your DNA, like 10 DNA samples.
Yeah.
Then you barcode, so you shear them, right? You need to shear to break the.
Yeah.
So then you can put these adapters and then make a full library, like for Illumina.
Yeah.
Or yeah.
Yep, I'm just gonna show you now the end of the website. So I didn't talk about that because we don't do it as much, but 100%. So there's two ways to do it. I'm on the Nanopore website actually. So they have what's called a native barcoding kit. So you can barcode without having to do PCR. It's just an additional like chemical reaction. So you ligate the barcode and then you ligate the sequencing adapter. And you can do, I think they have up to 96 different barcodes you could do. So they have a kit for 24, a kit for 96.
That's 8K. 8,000?
No,
No?
800.
800.
Yeah. So not as expensive. Not as expensive.
Okay.
And this is also, you have 96, and I think you can do it like 6 times if I remember. 12 reactions. So you have 96 times 12.
Okay.
So there's a lot, you can do a lot.
Yeah.
So this, and you can see here, they're showing, if you do no barcode versus the 96, it brings the price down from 600 down to $10. So you can do it that way.
Okay.
When I was in Oxford, I used to do that for like amplicons. So, there's no need to be doing a 111. We would just, barcode the amplicons, and then because the epitome already knows the barcodes, it would multiplex it for you as well. And then the other way to do it is you have, 2 other ways. You have this rapid barcoding kit, so it will do it really quickly. So it's a process called tagmentation. So it will fragment the DNA and at the same time it tags it with the barcode. So it happens quite quickly. So it only takes an hour. So that's another way to do it. And then you have the PCR version. So you can add it via PCR if you're doing cDNA or any PCR. Because you can still do PCR for this if you want. You don't have to. But you can add a barcode that way as well. So yes, you can definitely multiplex. It obviously all depends on how much data you need. So you would not multiplex like 100 if you need, a certain amount of data, but very flexible in that, 100%.
For environmental DNA.
That again?
For environmental DNA, eDNA.
eDNA, yes. So
You
yeah.
generally still need to PCR it, right?
Yes. Yeah. Actually, we are working with a company that has the bioinformatics platform for the bioprospecting, so taking ocean samples and making sense of it.
So for example,
But they don't do the sequencing, so they're going to work with us. So we're going to be doing multiplex barcoded eDNA samples.
So yeah, but for the metagenomics, if you want to sequence, like shotgun sequence everything,
Yeah.
for your mix of bacteria or whatever.
You can still barcode with a native barcode.
Okay.
Because you're gonna just ligate, basically just ligate to the end of everything.
Yeah, but and but for the sediment study, you are targeting microorganisms, right?
Yeah.
So there's plenty. It's
Just
far more than just the remnant DNA from the shark that passed by or whatsoever. So it's because you are targeted that you can do it straight away.
Yeah, exactly. Yeah. So I was just coming on here because I wanted to just double check, but with epitome, I don't think I have downloaded it. I'm just saying. I don't see it. But I think one of the workflows actually will, like I said, do the demultiplexing for you.
Okay. Additional question for the bed file. So when you want to filter, which could be great. For example, I want to do, so if I want to do phylogenes with 10 nuclear genes, I need to make my 10 amplification, my 10 single sequencing, my alignment, blah, 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 here. I can tell. So how much can I say that? Because I will have variant in the sequence.
Yeah,
So if, for example, I want to have sequence 10 or maybe 20, I don't know, but
yeah,
10 nuclear genes
Yeah.
or genes
yeah,
and nothing else, but for different individual, and then after the phylogeny, blah, blah, whatever. Can I tell what should I put inputs to do so?
yeah,
If I have, for example, one outgroup, so on birds,
Mhm.
to have everybody on board, so no, it's not human, but it's
yeah,
for example, you have the sequence of a bird, like a pigeon,
yeah.
but I want to have the colony from the Caribbean islands.
Mhm.
So I have just the reference from this species. I can put the sequence of the gene I know
Yeah,
in this bed file,
yeah,
and then sequence
just that.
just those,
Mhm.
and then I could multiplex by crazy,
Yep.
because I have just these 10 genes, and the other fragment, they will not be used.
So what happens is, so yes, you can do that. With the lab that we are doing our validation against in the University of Washington, they actually multiplex the human cancer panel. So they're only looking at 10 genes.
Mhm.
They multiplex, I think, 4 or 5 together and then run it. So you can definitely multiplex for adaptive sampling. Yes, that's why I love it as well, because you don't have to do any of that other like pull downs and libraries. The machine is doing all the hard work. You would prep it as if you were doing like the whole genome. So you just do the native sequencing, native barcode it, and then tell the machine, okay, this is what I want you to do, but it is no different. So actually for our prostate cancer study, what we're doing is one library prep. There's the same library prep, but we run it, because when we do the elution, we need 8 microliters, and we elute 32. So we can run it 4 times if we wanted to. So we take the first one and we do adaptive sampling of the genes we want. And if we see that they have a known prostate cancer gene, we don't need to do the whole genome to look for new genes because we found it already. But if we don't find one that we know about, we already have the library to run it for whole genome to find it. Now, the interesting thing with adaptive sampling is because it has to read some to make a decision if it's matching or not, you actually still get the whole genome, just at a very low coverage. So we found we did like 30x enrichment, you still got about 4 or 5x of the rest of the genome. You kind of see what's going on. So you still have more data than just those ones you're looking at.
Yeah, no, but you are not losing if you have variation between your reference and your actual sample.
No.
That's great.
Yeah, so that's something we're looking to do with our birds, our national bird, that's why we're doing this population study, is to find like, what are the genes that will tell us the genetic variation in the population? And then we can just run that panel and assess the health of the population versus the whole genome. What I will say, the limitation is that it has to be, or the pieces of DNA have to be around a kilobase long, because by the time it reads it and spits it out, like it'll be too short if it just reads the whole thing. So that's where like FFPE is not really ideal for adaptive sampling because they're all short pieces. But if you just do native, DNA from the bird or whatever, then you should be fine.
That's something.
Yeah, that should be absolutely fine. Metagenomics also a bit dodgy because it depends on how long those pieces are. We're gonna still try it though, because no one's really tried it, so we're gonna give it a go. With eDNA samples and see, because the project we're working with want metagenomics and they want metabarcoding. So we're saying, well, maybe you can try adaptive sampling for the barcoding versus having to do the PCR and everything.
Yeah, but for the barcoding, that won't be so efficient because generally you are targeting like 100, 200.
Right. That was what we were saying. So we need to figure that out. But yeah, there is that limitation on the length for adaptive sampling.
And eDNA is the problem.
Right.
The real DNA.
Yeah.
Is that's your DNA. So eDNA, environmental DNA is the DNA that is free, generally outside of the cell, but the organism that's are swimming in the water or on the ground left when they pass or they die.
Yeah.
So they are generally fragmented and they are short.
Right.
So for now.
So it might not work. That's the limitation.
That's great. For the bird, how much would it be for one to get one genome sequence, like one reference genome? What would be the cost for getting one?
So, do you have an idea how big the genome is, on a gigabase?
Yeah.
So we didn't know how big it was, it was bigger. We ended up doing 120x coverage for our wrap. We didn't mean to, we thought it was bigger than that. But we want to do if you want to do a new reference genome, we want about 60x coverage. At 60x times 1 is 60 gigabases. That's only going to be one flow cell. Looking at about $800 just for the sequencing. Not horrible.
And plus the prep.
That includes the prep.
That includes the prep.
Yeah. If you get blood, if you have blood, I mean the extraction is like $50. That's not that expensive. The library prep is about 100, and then the sequencing is the bulk of the cost.
And that could be after on the way down to here is the biobank and things like that in collaboration.
Yeah, so that's the conversation we need to have with standing up the biobank, because we're gonna have P48. That's what we're gonna put here at the biobank. So it will be there. We're gonna put a petabyte of storage and compute in the biobank as well. So there's no reason.
Project, I have samples.
There's no reason why like you can't do the extraction, do the library prep, and have it ready to just run on the machine wherever you are, and then just drive it down to the biobank, and then it's just learning on the machine. So you're not doing any of the prep there, because obviously they have clinical samples, but there's no reason why you can't like do everything up to that point and then just load it on the machine. And then, yeah. Because again, each of those positions are independent of each other. So we actually, when we were doing our breast cancer study, we were doing breast cancer samples, and then we had a coral sample sequencing as well. We were sequencing, because each one is independent, it's not messing with the next one. So that's the goal. I just need to get Stanley on board. And Jackie.
You have a question? No? Okay. Yes, I may have a last question. You have studied a lot of human genomes.
Yeah.
From the Caribbean.
Yeah. Not a lot, but some.
Okay.
We're getting there. Yeah.
You say that people from the Caribbean are more prone to getting some diseases than other people from the rest of the world.
Good question. I mean, I think from the existing epidemiological data, that's clear. We are like top of the world for breast cancer deaths, prostate cancer deaths, diabetes, obesity. We're just like #1 in all the bad things. From a genetic standpoint, I feel, and I'm biased obviously, but I think that is genetic. We did so far just breast cancer, but in Bermuda, we didn't test everywhere. Normally with breast cancer, you have BRCA1 and BRCA2, you probably have heard of. Those are the main genes that everyone looks at. In most populations, that's like 90% of the genes that's causing breast cancer is BRCA1 and BRCA2. In Bermuda, it was only 26%. We had 75% of our genes were not BRCA1 and not BRCA2. So what's driving the breast cancer is different, which means the biology of it is different, which means the treatment should be different, how it's handled should be different. I think that's part of the problem. We're copying and pasting what people are doing in Europe and the US, and then doing it here, but it's not the same cause. So I think that's part of the problem. So that's what the research we're doing should show. Right now, we've done 3 men so far genetically tested in the prostate cancer study. 2 of them have known mutations, not BRCA1, not BRCA2 already. So it's just like we need to not generalize or assume that we should be doing the same thing as elsewhere. I don't know if you have more. My sense is that, and this is gonna touch on like how we got here, right? You took us from where we were, you translocated us into a different area of the world, gave us different food. Like our body wasn't designed to be eating what we're eating, right? So that's a whole thing. And then on top of that, we don't have things designed for us. So we're just doing what everyone else is doing. So I think those compounding factors bring us where we are. But that's what we hope to find is doing the prostate cancer study. Maybe there's a gene we're missing. That is what's actually driving the high death rate in the Caribbean. So now we can test for it. Now we can screen for it earlier. And if you have this gene, you get a PSA earlier. And you're watching your prostate earlier rather than waiting. So I think I'm biased, obviously being a geneticist, but I think that's what it is.
Okay, very interesting.
And then, the other thing I'll say is the environment, right? So the epigenetics, which I'm really excited to get into. We haven't started the analysis yet, but the way that the environment is having an impact on our genetics is going to be different from elsewhere. We have, like in Guadeloupe, we have this pesticide, right, that was used on the banana plants. Like that has a direct link to higher incidence of disease, and we can test for that now. So you have all these different factors as well to think about.
Okay. When you sequence the genome, you say that basically you have the epigenetic marks straight away, or you need a specific prep, or is it
straight away? Well,
straight away?
You have to press a button when you do the sequencing and say you want modified base calling on. It's just literally like tap on and then it's there. And in the workflow that I showed you that I was doing, it actually also calls all those variants, all the epigenetic variants. So for the breast cancer and everyone we've done, we have the file that has, and again, you can probably run it. So we need tools to look at it and figure it out, but we have all the data. And same for the birds. So for all the birds, we have epigenetics as well. So we can see, and we've actually have a few birds that we're tested in like 2019 and then in 2024. So you can see if there's any difference between
Same individual,
because they're all tagged. So we know it's the same bird, just different times. You can see if the epigenetics has changed. Because our national bird, is a seabird, and it flies away for years, and then it comes back to Bermuda. And it only breeds in Bermuda. So we can see what all the flying around the Atlantic is done. So yeah, but you can it literally is no different in terms of prep or anything. It just has to be native. You can't do PCR. But you just have to press a button and then you have it. You get it at the same time. Yeah. Otherwise, if you wanted it, you'd have to use a different technology and different samples, double the cost. So it's more efficient this way.
Well, thank you so much for giving us this presentation and answering so many questions. Very interesting. We could stay there for many more hours, I think. Thank you again. Maybe we'll cross path again one day.
Yes, I will be back. Thank you so much.
Thanks a lot.